Okay, so we're going to be talking about the DNA replication process on this right side of the image, this lagging strand here. This is part 2 of a 2-series video on DNA replication. So if you haven't gone over the first video about leading strand synthesis, I would recommend that before we jump into lagging strand synthesis, simply because there's a couple more components here, but some of them use the same components, like DNA pol 3 and beta clamp, as well as, this all kind of starts from the same process up here. So just to review, the lagging strand is going to come from the very beginning of this process with topoisomerase, which we talked about in the first video, but just to give a quick overview, DNA gyrase or topoisomerase is going to be responsible for keeping the DNA from supercoiling, so making sure that it's loose so that we can hit it here with DNA helicase, which will unwind or unzip the double helix. Now, here's our first difference between leading and lagging strand. So lagging strand, we're going to see up here primase. Primase is going to make this RNA primer that's here in dark green, and this RNA primer is going to be, like noted here, at the beginning of an Okazaki fragment. We're going to go over what an Okazaki fragment is a little later, but nonetheless, we're going to make this RNA primer, and it's RNA, not DNA, which is kind of weird, right? We're making DNA. These teal strands are newly synthesized strands of DNA. Why is there an RNA primer here? Well, RNA primers are going to be really helpful for when we're looking at getting some other enzymes, like this purple DNA pol 1 and this dark, like burgundy color, which is DNA ligase. This primase, or the primer that comes from primase, is going to be really helpful in flagging down DNA pol 1 because there are differences in the way that the leading and lagging strands are synthesized. So if you look here, the leading strand is kind of going in towards this kind of area here, this replication fork. And if you look, it's flipped kind of up and around and upside down. But what's really happening here is that this parent strand is looping around here, coming around. It's actually being synthesized going away from the replication fork, which is different than the leading strand. And there are differences in the leading and lagging strand synthesis because these two parent strands are being synthesized in opposite directions, because if you remember, DNA is complementary to each other. These two strands of parent DNA are complementary to each other. So one is in the 3' to 5' direction, and the other is in the 5' to 3' direction. But there's, we can only synthesize in the 5' to 3' direction. So we're reading only in the 3' to 5' direction, meaning that these two strands are opposite, and so we're going to have to synthesize them in opposite directions. So leading strand is being synthesized in towards replication fork. Lagging strand is being synthesized away from the replication fork, which leads us to these differences. So first, we're going to use primase to make that RNA primer. We saw, like in leading strand synthesis, these little purple dots. These are all SSBs, single-strand binding proteins, that will keep the DNA from binding to itself after going through helicase. Now we're also going to go through DNA pol 3, which is going to synthesize our new strand of DNA after this dark green primer made by primase. We're going to continue out and down this pathway here, and what we're looking at is, so we have some of these elongated strands of newly synthesized DNA, but there are still primers here, which leads us to the Okazaki fragment. Okazaki fragments are kind of short DNA fragments that are created by DNA pol 3. And because we're synthesizing down this direction, if you think about it, when we're synthesizing the leading strand, it's going in towards the replication fork. It's continually being fed new DNA of the parent strand because it's going in towards. So that's different than the lagging strand, because the lagging strand, the replication fork is producing DNA, a single strand of DNA, to be synthesized in this direction, but we're synthesizing in this direction. And that can be a little tricky to wrap your head around, but basically we're going opposite the direction that the replication fork is outputting. So we have these Okazaki fragments because we're having to kind of synthesize and then go back and then synthesize some more and then go back and fill in these fragments, these RNA primers. So what happens is these primers are like a flag to DNA pol 1. And they say, hey, I'm RNA, please come chew me up and put DNA here where there should be DNA. So that is what DNA pol 1 is there for. It's going to chew up the RNA primers and fill these gaps with DNA. And we're still not done yet because we have fragment, and now it's all DNA, but we need to glue it together, which is exactly the job of DNA ligase. DNA ligase, like I noted here, is responsible for going and gluing these Okazaki fragments together. So that is why there is a leading strand and a lagging strand on DNA replication. Just because you can only replicate out in one direction, and when you're synthesizing, you're, going in towards that feeding of DNA, or you're going away from it. So you need to kind of, go back again, again and again and again, and yeah. Alrighty, that will be it for this video. If you have any questions or comments or suggestions of what you want to see next, please let me know. I am totally open to hearing those, and I hope this was helpful. I'm probably going to do another kind of overview of DNA replication, just to really help you all understand it. So, alrighty, I'll see you in the next video.